launching the PowerPoint. Initially, I made to talk about lupus in general, but I think it's a good forum for you to show you a little bit where I came from, what I have done, and what actually made me come here. So, brief introduction, I'm going to talk to you about my education, O level, that's grade 9 to 12. A little bit about A level, grade 13 to 14. Undergraduate, graduate school, postdoctoral work, career in biotech industry, and research institute, and then finally here. And so when we reach here at the Oklahoma Medical Research Foundation, that's where we'll talk about lupus. That's the last work I did. I believe any one of us here who is a scientist, our career paths have been dependent on other family members, other people you met along the way, and also events. And I'm going to discuss, as I go along, I'll mention some of these. So, before I start, I'll show you where I come from. If I didn't go to school, this is what I've been doing. Actually, my father was a fisherman and my parents never went to school. This was everything that I knew growing up to actually to college until I came to the US. I keep telling my kids, I look like this at that age. They don't believe it. Anyway, I come from Africa, a country called Kenya, follow the dot. And in Kenya, I live in the western part of Kenya in Lake Victoria. Lake Victoria, this is the source of River Nile. And if you explore it a little, this is the island I come from. It's called Mufangano Island. So the O level, the grade 9 to 12 in our system, at the end of grade 7, you do a national exam. If you pass that, you are admitted into what you call the secondary school. Secondary school is what you call here high school, grade 9 to 12. And at this point here is where I met somebody who actually changed my career up to now. And it's the person who made me come to teach. He was the headmaster of the school, and since I came from a very humble background, I didn't have school fees. We pay school fees, grade 12 to 9 to 12, you pay school fees all the way to the university. I didn't have school fees, but he kept me in school for 4 years without fulfilling the fee requirement. And one day, towards the end of my 4th year, he called me in his office. We prayed together, and he gave me advice. He told me, I can be able to do anything I want, except if I self-destruct in the future. And I'll tell you how that thing has lived with me throughout my career. After grade 12, I was offered, did another national exam, offered a slot at high school, also around here. After grade 14, I went to the university for 3 years. The university I went to, actually that year, the people who took grade 14 exam were 54,000. 18,000 had the qualification to be admitted to the university. Only 4,000 got the slots. When I was in high school, grade 13 to 14, you only choose 3 subjects. So my subjects were math, physics, and chemistry. And the physics was my best subject. But when I went to the university, I had to be practical. Practical in the sense that I'm the second born in our family. Our older brother stopped after grade 12 to go to work so that he can help my parents pay our school fees. So it was now my opportunity to get a career that is going to pay well, so I can also educate my remaining sibling. We were 8 in total. So I chose chemistry because there are a lot of industries in Kenya, Firestone, Coca-Cola, and all these pharmaceutical drugs are doing manufacturing in Kenya. So if I got my chemistry degree and a good grade, I was guaranteed a good job. And that was my goal when I went to college. And the key is for you to get a good chemistry, you have to go through 3-1-1. So the first year, you take 3 subjects. So I did geology, math, and chemistry. Then out of 400, they only take 20 people to do chemistry. That's why it is called 3-1-1. So the first year is 3 subjects. Second year, you only do chemistry. Third year, you only do chemistry. And 20 people, by the end of second year, everybody has a job before they finish. That was my goal. So after 3 years, the end of my third year, one of my professors called me. His name is Dr. Mideo. And he called me in his office on Friday and said, "What is your plan?" I said, "My plan," I gave him the field that I just gave you. I want to get a good job, take care of my family. I'm in his office talking to him. I'm very excited that I've gotten a job. I've gotten about 5 offers already. And he is busy doing his things. And when he stands up, he says, "You are going to graduate school." I say, "What are you talking about?" He said, "Yeah, you are going to graduate school. And for you to go to graduate school, you have to take directed research projects." And I told him, "I've already enrolled for classes for next year." He said, "Yes, I know. And I can't drop one now." He said, "No, I just intercepted your enrollment form and I added that one." And I said, but we are only go to school Monday to Friday and all those slots are filled." He said, "Yeah, you still have Saturday and Sunday. You'll be doing that during those 2 days." And remember what my headmaster told me? You can do anything you want unless you do what? Self-destruct. That thing was playing in my head. So I stopped arguing. And so he said, I said, "When do we start?" He said, "Tomorrow." That was Friday, tomorrow. So he brings a file from his drawer. I said, "Read these things tomorrow. Come to my office on Saturday at 9. I'll give you a project and you'll start." And at the end of so during that time, my career changed. I started looking into graduate school. So I look in the board. Boston University had left this card asking for graduate students. I filled one. I was late. But so the chairman of our department called them and said, when they sent the application, told him my situation, and they said they're going to waive everything except one thing, is I have to get first-class honors to get admission. It's a piece of cake. I talked to my father. My father said, "You better get that or don't get home. Don't come back home." So everything, I came here in August 1995 at Boston University, and I started working on mechanism of cancer therapeutic drugs, mostly nitrogen master, under this wonderful gentleman here, Dr. Edward Leckler and Standis Hassman. Dr. Dory Grunberg was my fellow graduate student. Nitrogen master was used in World War II as a chemical agent. And during that time, what they found out was it really affected the bone marrow and the white blood cells. So they started studying if they can use that to treat lymphomas, things like leukemia. And actually, it is now used standard chemotherapy for a lot of cancers, Hodgkin's, non-Hodgkin's, chronic leukemias, and other cancers. The goal for me was to find the mechanism of action. It was known that it was reacting with the DNA, but which one was the correct adduct for it to work, to stop the DNA proliferation? So my work was to calculate nitrogen master. This is the structure. You have chlorine on either side that are very labile. So if you have a nucleophile, attack this and this leaves on this side, this one also a nucleophile, and it is found to be reacting with 7 position of guanine. They have a nitrogen there with two electrons. It can donate the 2 electrons following the ring rearrangement. To cut the story short, at the end of my 5 years, what we found was the linkage was the adduct that was inhibiting the DNA replication was the interstrand, and it was attached to 7 guanine and 7 guanine on the other strand. After that, the result from that work actually improved some of the nitrogen mastered derivatives like cyclophosphamide and isofamide, which are actually still being used today as chemotherapy for leukemia. After my graduate school, my older brother was diagnosed with AIDS, HIV, and some of my friends were actually sick at that time. I decided to do something else, to maybe make an impact in the AIDS research. So I contacted Dr. Flossie Wangsall. She was at NIH at that time. She was in Bob Gallo's lab. Gallo was one of the people who discovered the HIV. And she was just moving to the University of California, San Diego. I didn't think I had a chance because I'm a chemist. They need a virologist. And everybody in the world wanted to work with her. Fortunately enough, I had got good recommendations from my bosses. And she hired me. So I moved to the UC San Diego, California to study virology and mostly HIV. And my project was to gene therapy for HIV using the new technology at that time that was ribosome. A little bit back a little on ribosome. Ribosome, all along people had thought that all enzymes are proteins until Tom Cech, who was a professor at University of Colorado at Boulder, discovered that RNAs can also act as enzymes, and hence the ribosome. And actually, he got a Nobel Prize for that. So, as you young people who want to go to biotech industry, if you develop a drug or discover a drug, you have to know the mechanism of action of your target. And my target in this case was HIV. For me to inhibit, to control HIV, I have to know the life cycle of the virus and try to intervene in some of the critical parts of the virus life cycle. So HIV, the first thing it attaches to is CD4 receptor, then it rolls into the core receptor. After that, it gets internalized into the cell, and then it spits its genetic material. HIV is a retrovirus. That means its genetic material is RNA. So you have RNA here, and this RNA here, what it encodes 2 major proteins. That is the reverse transcriptase and the integrase. The reverse transcriptase makes the RNA into DNA, and the DNA is transported into the nucleus, where the integrase integrates the HIV genome into the host genome. After that, then it starts replicating, and it bursts out, and you have a lot of the viral titles. My goal was to, using the ribosome, was to inhibit the RNA, to break the RNA once it has been injected, to break it before it gets replicated using the ribosome. So this here is the HIV genome. You have the LTR, the long terminal repeat on either side, and then you have some of the major proteins. This is the GAG, which makes the casing envelope, and this one is the Paul polymerase that reverse transcriptase and other proteins. So my goal was to leave the RNA at this position in the GAG, and if you do that, then it's not going to be available for transcription, replication, and transcription. And the ribosome I was using was called a hairpin. This was developed by Dr. Hampel. He was at the University of Iowa, and I had to do this. This is the HIV RNA, and it binds to the the ribosome, and at this position here, it gets cleaved. And so what you have the ribosome, which acts as an enzyme and breaks the RNA, the HIV RNA, and makes it infectious. So what I did, since this gene therapy, what I wanted to do, or what I did, was to clone that ribosome into a vector. A vector, in this case, is a vehicle for transporting your genetic material into the cell. And once it's in the cell, it's going to be, you have a promoter here, it's going to be replicated, transcribed, and you're going to have RNA, which is going to bind to HIV RNA and cleave the RNA, and hence stop the HIV replication or production. This work worked very, very well. And actually, just before I left, Flossy Wangstad started a company called Immusoft, and they use this technology. Actually, they still use it for other viruses, not for HIV anymore, because we have now drugs. After that, I got my first job. All along, I wanted a job. So I got my first job in a company called Triplex Pharmaceutical in Woodland, Texas, that is just north of Houston. And the technology they were using there is TFO, triplex forming oligonucleotide, or antisense technology. And since I was the HIV expert now, away from chemistry, the technology was using this platform. You know, the DNA is double-stranded. And for the DNA to be replicated, you have promoters and other factors that bind to the DNA, read the DNA for replication, and eventually for transcription. The TFO, or the triplex forming oligonucleotide, can bind to some of these areas. Some of these promoters are purine rich. Purine are where you have guanine and adenine. So in those areas, the TFO, the oligonucleotide, can bind and form a triple helix. When they form the triple helix, they compete with other transcription factors and prevent transcription or arrest transcription elongation. And by doing that, you are actually killing the virus before it can replicate, before it can form RNAs. And so this is sort of how it binds. So you have the two strands, and then you have a third strand. And this is how you have one is Hoopstein, the other one is Weston Creek kind of hybridization. So we were using this for HIV, and we designed some oligo after a lot of trials. We got one that was very, very effective. This one is called IC50. IC50 is the inhibitory amount that is required to inhibit 50% of the virus. So the lower the number, the better the drug. And so everybody was happy. We have a compound that was designed for HIV as a triplex forming oligonucleotide. We have sort of authenticated the platform. Guess what? We start doing more research to it. We find that it was not acting through that mechanism that we thought. It was still potent, but this compound, this oligonucleotide has a lot of G's, guanine. And in solution, it forms a tetrad. And it is this tetrad here that actually binds the HIV integrase, the other second enzyme, the enzyme that once the DNA is formed and it has moved into the nucleus, integrates the HIV genome into the host genome. So by accident, we found that. You're still excited because you found something. This compound was actually taken through clinical trial. Just briefly, when you do drug, after discovery, you do what you call phase 1. Phase 1 trial is for, you do animal models. The animal models, you want to see how the drug is it toxic to the animal, and how much can it be tolerated. So it helps you giving you the dosage. And then you do what you call phase 1 clinical trial. But phase 1 clinical trial is where you take naive patients, non-sick patients, and try to see if how much can they tolerate, and you take their blood to see how long does it take the drug to be in their blood system. And that will help you with the dosage, how often you dose, and do you have the critical level to inhibit your target. So it went, and the company sort of ran out of money, but this thing was taken by another company, and this technology was actually improved, and one of these drugs is in the cocktail of one of the HIV drugs that are being taken right now. Triplex was bought or merged with another company called Aronex. In the Aronex, I worked with Dr. Tom Wallace, now in the pharmacology department. Aronex had a liposome technology. Liposome is a phospholipid vesicles. If you have dry lipid form, given appropriate weight, molecular weight, they spontaneously form vesicles. And this happens because, as you can see, they have hydrophilic tail, the water-loving tail, and they have hydrophobic tail, the water-hating tail. If you put them in water, dry in water, this is what you're going to end up forming. And when they do that, they give you a pocket here. This pocket here you can use to enclose any drug you want. And they can deliver this drug any place. And since they have phospholipid bilayer here, they can actually interact with the cells and deliver the payload inside the cell where you want it. So this is used to, first of all, transport drugs that cannot be transported alone because maybe they are too soluble in water or they are too large. And so we use that technology. There were two drugs that one of them was called Atrogen. It's a cancer drug. It's all trans-retinoic acid, ultra. This is the one that a modification of this is what we call a face cream. It's a very good anti-leukemia drug, but it cannot be delivered as such because it's very soluble. This is the oral bioavailability. That means if you inject it straight, it's going to bind to blood proteins. And so it's not going to get delivered. You're not going to have enough concentration. And if you want enough concentration, then you have to add a lot, which means you're going to have side effects. So what we did was and this is the structure, was to put it in that liposome, and we can now deliver it to the target cell in good concentration, which is non-toxic. This drug here is in the market, and actually it's being used right now for treatment of acute lymphocytic leukemia, where there is no treatment particular for the kids. The other one was antifungal. Fungus, you have topical fungus, you have athlete's foot and other things. Those ones are topical. But people who are immune depressed can get what you call systemic fungal infection. If you get systemic fungal infection, they are very deadly. I think a typical example will just happen here. Maybe a few months ago, the people who got the contaminated drugs from that lab from Massachusetts. So systemic fungal infections are very deadly, so they needed a drug. And nystatin, which is a topical, is a very good, but it cannot be. This is the structure, and you see there are a lot of OH. Once you have this OH, that means you have problem with solubility issues. And at that time, the only one that they have was amphotericin B, which is also a big one. So we encapsulated this in our liposome technology and did a lot of clinical trial. And actually, this one is now being used for treatment of systemic fungal infection. So in 1997, I moved from Texas to Oklahoma to join this company called Zymetex. In Zymetex, I was to head their virology department, dealing with mostly influenza. viruses and RSV. And our VP of research was Dr. Shimoshaki, and some of these are my colleagues that we work with throughout. And what we did was we developed a diagnostic kit for influenza, which would be able to detect influenza A or influenza B within 30 minutes. So, as usual, for you to design a detection kit or a detection or a drug, you have to understand the what? The life cycle. So you have to understand, this is the influenza virus. Influenza virus has two critical proteins that we were interested in. One was neuraminidase and hemagglutinin. What happened is that hemagglutinin here binds to the receptor, which allows the virus to get internalized. And once this is done, then it bursts out of the cell. This one here is the neuraminidase. What happens is, when the virus bursts out, it's still attached to the cell. The hemagglutinin and the cell receptor are still attached. Neuraminidase is the enzyme that cuts the hemagglutinin from the receptor to release the virus. So if you target this enzyme here, you can be able either to treat influenza or to have a diagnostic kit. And actually, just all the, Tamiflu, Relenza, actually they are inhibitors of neuraminidase. And I'm going to show you the structure why the other one can be taken as a tablet, the other one can only be taken by IV. So what we did was to target this, and to target that we have to understand its natural substrate. And the natural substrate is what you call N-acetylneuramidic acid or salicylic acid. So we know that what it cleaves. This is what is attached to hemagglutinin, and it cleaves at this point here. So what we did was to engineer, improve, or make derivative of that salicylic acid. And what we added a methyl group here, and at this end here, we added here what you call something that's a reporter, this group here. So what happened, still this thing is still available for neuraminidase, it's still recognized as a substrate. Neuraminidase cleaves this. This is what you offer, it cleaves that, and what happens, a specific cleavage. And what you have is you have this in solution. This one you treat with a chemical, which allows it to what? To hybridize. As a single molecule, there is no color. If it is hybridized, it is now a blue indigo. So essentially, if you have virus, then the virus is going to cleave this, and you're going to, if you have blue color, you have virus. If you are not, you don't. So I'll show you in just a minute. So this is the kit that we have, and this is, you take a swab from your throat, from your tongue. You put that swab here in a small tube that has detergents to break the virus, to release the enzyme. Then you drop that into a bottle that contains your substrate. You incubate that for 30 minutes. You take it out, put the stop solution. The stop solution is the one that is going to, bind the two neuraminidase, the substrate. And what you end up with is, if there is no virus, that is negative. If there is a virus, you're going to see a positive. And actually, what we did was, that was a 30 minutes. We wanted to make it a 5 minutes. So you go to the doctor's office, in 5 minutes, you know whether you have virus or not. So this is our original chromogenic substrate. Now what we have was we now attach the substrate to a chemiluminescent compound. So essentially what you do is you end up with, this was a Polaroid. you mix your solution, put them here, and in 5 minutes, this has a timer, and in 5 minutes it spits out your Polaroid instant negative. If you have a virus, you're gonna be white because there's light gonna shine, and there will be no light. This one is we only had a prototype because at that time the company was sold to a group in New Jersey, and I did not move. But I was just going to tell you, this is the structure of Tamiflu. This is the structure of Relenza. They are the same thing. The reason why Relenza can only be administered by IV is because you have these OH groups here. This was actually developed by a small company in Australia, sold to Glaxo, and this was actually developed by Gilead, which is now sold to Roche. And you can see what the Roche people did was to add here methyl groups instead of hydroxy group, and this makes it orally bioavailable as a drug. This one cannot be, has to be supplied by. Okay, so when the company was sold, I had a decision to make. Should I go to the company to New Jersey? My kids were still young. They were going to high school. So I was outvoted. And guess what? I have 3 girls and 1 boy, and including my wife, so I had no chance. So I had to find a place, and I had a good friend who was the chairman of Dr. John Hurley, who was the chairman of the Department of Arthritis and Immunology at Oklahoma Medical Research Foundation. And for all along his career, for about 18 years, 20 years, he wanted to work on, he's been working on lupus genetics. He said after doing his MD, PhD, he had a friend who was stricken by lupus. So that was his career. It was to actually shed some light to the disease. So he convinced me to join his group. And this is my people in my lab that I had for the 5 years that I was there. And this was because you have to collect samples, lupus patient samples. They had accumulated about 10,000 patient samples from all over the world. So let's go talk about a little bit of lupus. The College of Arthritis and Rheumatology classify lupus as uniform rash. There are a lot of phenotypes of the way you can also discoid rash. I'm going to show you pictures of some of it. Photosensitivity, oral ulcer, arthritis, and kidney involvement. This is the major one sometimes to some people. Some people also have hematological disorder, and they all have this thing here. Essentially, lupus is you're producing your antibodies to yourself. You see, when you get infected by a virus or a bacteria, your body recognizes that as foreign and produces antibodies to counter the foreign. In lupus or any other autoimmune diseases, you are actually producing antibody to yourself, and so you start fighting yourself. And they produce a lot of interferon, typically affects women in the childbearing age, 18 to 40 years. We're going to see a few of these. And the signs are fatigue, non-erosive arthritis. What this means is you find you have joint aches, but when they do testing, arthritis, you erode your cartilage. In lupus, you don't erode. There is no erosion. So there is no deformation. So these are typical manifestos. This is for the kidney, and you look, it manifests in different ways. And what they have in common is if you take their blood and cells and you treat them with antibodies, this is what you see in your confocal microscope. The product of autoantibodies. And if you follow patients people who have lupus, you can actually see the titers of those autoantibodies rising with time. So you have different one here. This is the autoantibody, the anti-Ro, this is different for different human proteins. And so with years, this one here is where you start the onset of the lupus. You start seeing the disease manifesting. The ratio is female to male is 10:1. So it is more prominent in women than men. And 1 in every 1,200 Caucasian women, and it's more prevalent in women of color. So it's more common in African Americans and mostly African Americans. And the genetic implication. Why is it a genetic disease? And this is how you classify this kind of things. Is when you look at families that have one person has lupus, 10 to 20% of that family tree is going to have lupus compared to a general population. The other thing is if you compare siblings, the ratio is about 20 to 1. If you take twins, if you take monozygous twins, that is the identical twins, the chances is about 34%. Let's say if they're identical twins and one of them has lupus, the chances of the other one having lupus is 34%. But if you take dizygotic twins, that is a non-identical twins, the percent is only 3%. So this implicates there has to be a genetic component to the disease. So if you have a genetic component, then you can have to study a little bit of genetics. And the 3 methods that have been used to study this genetic disease is what you call genome-wide linkage analysis. This was the original way of learning this, doing the genetic of the disease. The linkage is like this. If you have a family member that has the disease, you start following the family tree. And so you collect DNA from all those family members, the ones that have the disease and the ones that don't. And if you do genotyping, you'll find if there is a polymorphism that is common to those people with the disease against those people who don't. Then there is what you call a candidate gene. The candidate gene is where you know the gene, you want to study it into detail and find out which polymorphism, because genes have different polymorphism. You want to find which polymorphism is actually associated with the disease. The other one, which is now the darling of the trend right now, is what you call the GWAS, genome-wide association. This one actually, what it does is it collects all the lupus-affected people compared with non-affected people from everywhere. So it is not a family associated. It's just you try to have DNA from lupus patients and non-lupus patients and do the genotype and compare which one is, which polymorphism is so associated with affected people compared to unaffected. And this is what we did in our lab for the 6 years that I was there. So let's define what we call by polymorphism. The one that we used a lot was this one here, called the SNP, the single nucleotide polymorphism. Imagine this is one strand of the gene, and you have an A here. If you look for somebody else, you have a G here. So the only difference is one nucleotide. This is what you call the other polymorphisms are insertion. You look at this strand here. Here you have added 2 A's here. So instead of having this one now has moved, so these T's are this way. So this is called insertion. The other polymorphisms are inversion. Inversion, you look at this strand here, you have TCA and ends with A. On the other hand, you have the same gene, but this one is inverted. The one we used a lot was the SNP. And there are a lot of very powerful genotyping platforms around. The one we used was a machine made by Illumina. They are somewhere in California, and essentially they use the PCR method. So what you have is you have the whole genome DNA, you break it down, what we call fragmenting, into small pieces. Then you have a probes, you do the PCR, and the PCR will amplify all those small fragments. And what you want is to do PCR again with the probes. This probe, so let's say what you have is, if there is T here, so this one here is the PCR product from your fragmented genome. You separate them and then you add the probe. So the probe here, one of them ends with a C. If there is a T here, this one will not complete, so you end up with that fragment, and so on and so forth. And once you do that, you put them in this slide here. This slide here has some probe captures. That's going to capture some of this. So it can capture T, TT, GT, and all those. And once you have them here, you put them in sort of a robot. It does the cleaning, the hybridization, everything. And once you're done, all you have to do is to put them in a oven and dry them. And once you dry, you put them in this machine here, which is the scanner. The scanner scans this thing. I mean, when we started, we had one that was actually scans 12 of these every 4 hours. That was too slow. So we bought this big one here. This big one here, we have a tray. And in 12 hours, you have scanned over 1,000 of this. So your genotyping, actually, the power of your genotyping went exponentially. And so what you look at, every slot of this, when you look at it, when it is scanning, it is something like this. And all this information is fed into a computer program. The computer program now, because you have affected, SLA affected patients and non-affected patients, you do all the genotyping. The computer will now, because you tell the computer these are the affected, these are not the, these are the unaffected. The computer now compares the SNPs difference, and then we leave it for the statistician to clean it up. So let's say, for example, you start with 478 unrelated cases. These are, you don't want to do families here. You take unrelated cases, people who are suffering from lupus but are not in relatives to one another. And then what the other thing you take is the SNPs of you make so many SNPs of those. And then you have control. Always you want control to be more than the cases. You clean them up, and eventually you end up with the good ones, 431. You started with 478. Some of them had some abnormality that have been cleared off. So you end up with 431, and this for control. And after the statistician crunch the numbers, they give it to laymen like us to look at it. And it looks like something like this. So you have all your chromosomes, 1 to 22 and X. Actually, you should also have a Y chromosome here. And what you look at is, these are the p-values, the probability of. The higher the probability value, the better the association. So everything under here, P is equal to 6, is noise. So you find all these ones are individual SNPs. These ones are not. But anything above this line here are actually associated genes, associated SNPs. And you find where let's say here is HLA region, and you have some other genes. So based on this work, a lot of genes have been associated with SLE, with lupus, and you have RA5 and all these genes. Some of these, they are genes that are associated with the immune function, and some of them have no relevance to immune known yet. And this is just a small number. They are now actually, I think there are over now 50 genes that have been associated with lupus. The question somebody can ask, why so many genes? You remember when I started, SLE, for some reason, manifests in people differently. There are people who are photosensitive. There are people who are having kidney failure. There are people with Marie-Rust. So because of these phenotypes, they have not been able to tie down an individual gene responsible for the disease. And that's the gist of the work that is still going on. So with all the information we have right now, this is what people who are genetically predisposed, and because lupus does not start, you don't know, it cannot. One thing that is now apparent, if a male has lupus, the chances that of its offspring having lupus are very, very high. So if you are genetically predisposed, and this can be triggered with so many things: environmental factors, smoking, drinking, stress, anything. Sorry, what did I do? Okay, so you start developing autoantibody, and once you have autoantibody onset, then you start having pathological injuries. You have now the clinical disease, and you start presenting SLE. So in summary, there is genetics, which is sex, race, and about 35 genes now, environmental, smoking, and sometimes viruses also. The etiology of the disease is still not understood. So unless it's understood, you cannot develop drugs for it. And currently, there is no cure, but these are treatments. And they are treatment for different phenotypes. So some people might respond to just NSAID. Some people will respond to anti-malarial drugs. And nobody knows the mechanism by which this really actually. And some people even methotrexate. Methotrexate is a drug that has a lot of quirks, and nobody knows how it is used for treatment of cancer. It is used for treatment also of arthritis, and it also has a function here, but nobody knows how it works. Immune suppressant can also be used. So after 6 years, I still remember what my high school teacher told me. I found that in around 2009, he died. And when he died, I went home and I went to the funeral. So it seems every student that he had touched had been coming every time. So I go there and the wife tells me, "Oh, you are one of his students." I said, "Yes." "Oh, they have been coming here almost every year." And made me realize that if I die now, will somebody really remember me apart from my kids? In other words, he touched me, and there's no way I can repay him. The only way to repay him is to pay it forward. And to pay it forward is to teach somebody like you. And so it was a big pay cut, but I had a friend who convinced me that you'll survive. If you come to teach, you'll survive. I've survived for 1.5 years, so I think I can survive. So what I leave you with today is you don't know when you'll make an impact on an individual. So try your level best to also shape somebody else's career. You have friends whom you know they have a potential, but you are afraid to tell them. Tell them. They might be upset with you at the moment, but later on they might think that, my course of action changed because so and so talked to me. They will never tell you. They may not tell you in life, but you have touched somebody. And my professor at the University of Nairobi, the one that I argued with, this is what he told me when I was trying to stop me from arguing. Don't let your attitude insult your intelligence. In other words, don't ask why me. Ask how can I do it, and can somebody help me with the tools? Because if you start asking why me, if you start to playing victim, there is nothing you will do. The days will come, the years will come, and guess what? The years don't care whether you're a victim or not. So if you want to do something, do it. Do it to the best of your ability. My father used to tell me, because he was a fisherman, and he kept telling us that I want you to be better than I am. And I don't care what you do. Even if it means you cleaning the toilet. Clean that toilet so good that if somebody sits on that seat, they forget everything they were thinking. They say, wow, this guy must have done a wonderful job. And when you leave that place, go to another. In other words, wherever you are, do your best. If you walk away, there are no regrets. Thank you. Thank you, WiSE, for giving me this opportunity. And thank you, gentlemen and ladies, for showing up. I'll take any questions. Yeah.
Why do you think that lupus is more common in women than men?
Those are the cases. They are more prevalent in women than men. And actually, I forgot, because of time, they even at the beginning they were thinking that it was chromosome linked, X-linked. And people started doing maybe it is estrogen. And those studies have not found out. But what is known, maybe people don't know. People are XX and XY. But there are people who are XXX, triple X. Those people are more susceptible to lupus than people who are XX. So the severity is XX, triple X, XX, XY. There are people who are also YY or XXYY, XXY. So they don't know, and that also it is during the 18 to 35, the childbearing age. So they think maybe there is an hormonal which becomes a trigger effect for that, and men don't have that.
So men who are XXY are also at a higher risk?
Yes. And you don't want to be there.
Would men XXY be at higher risk than the average woman XX?
They are actually almost equal. They are almost equal. Thank you. No, thank you.
Alex. I could stand here and listen to